Pranita, welcome to all.
Okay,
And let us start.
Okay, sir. So, good morning, one and all present here for this lecture series number 4, which is based on the theme Indian speaking from
1 minute,
abroad.
1 minute, Pranita. I request all to mute your mic, please. Everyone except Pranita should mute your mic. Okay, proceed.
Okay, sir. So good morning to one and all present over here for the lecture series number 4, which is based on the theme Indian speaking from abroad. So on behalf of Shivaji Science College, Congress Nagar, Nagpur and Microbiologists Society, India, I welcome you all. I welcome Honorable President of Microbiology Society, India, Dr. Arvind Deshmukh, sir. I welcome Honorable Principal, Shivaji Science College, Congress Nagar, Nagpur, Professor Mahendra Dhore, sir. I welcome National Coordinator of Microbiology Society, India, Dr. Patankar, sir, and all respected participants, as well as the most importantly, our eminent guest speaker, Dr. Gokul Shankar Sabesan, sir. So he is from Malaysia and he is delivering today's lecture. So on the occasion of 124th birth anniversary of Dr. Punjab Rao alias Bhausaheb Deshmukh, founder president of Sri Shivaji Education Society Amravati, we are conducting this lecture series in collaboration with Microbiology Society India. So let us start our today's guest lecture, which is based on the theme Indian speaking from abroad. Topic of today's guest lecture is how to make microbiology teaching and learning incredibly interesting. So let us introduce our today's guest speaker. Yes, Dr. Gokul Shankar Sabesan sir is currently professor and head microbiology department, Faculty of Medicine, Manipal University College, Malaysia. His expertise in his doctorate in the field of microbiology, armed with an extensive career of more than 14 years in research and a distinctive expertise of more than 17 years as faculty in microbiology for the postgraduate and undergraduate students in India and Malaysia. He has received many awards and accolades in his honor. Received Young Scientist Award in recognition of outstanding contribution in the field of medicinal plants research at international level in 2011. He has received SAB Award of Excellence in Food Safety in 2012. He has received Alumni Achiever Award in 2015. He is the recipient of 2016 Award of Excellence in Food Microbiology. He is the recipient of Best Teacher Award in 2019 from Microbiology Society of India. His expertise in QC and QA aspects of microbiology. He has worked as a principal investigator for FRGS grant in 2019 to 2021 in Malaysia. He has completed he has guided 3 PhD candidates from India and 4 MSc students from Malaysia. He has served as a lecturer, then senior lecturer, and now currently as a professor in Faculty of Medicine, Manipal University College, Melaka, Malaysia. He is the preclinical coordinator as well as deputy dean in preclinical and deputy dean in student affairs and alumni. He has served as a teaching microbiology and immunology to MBBS and BDS students. He is research supervisor. He is the member of many professional bodies. He is the member of organizing committees of the international preclinical quizzes and other organizations. He has received SAS Award of Excellence in Food Safety. He has also received Excellence Award in Food Microbiology. So very much thankful to you, respected Dr. Sabesan sir. You have achieved many more things in your career and we are very much thankful to you. And On behalf of Microbiology Society India and Science College Congress Nagar, Nagpur, we are very much thankful that you have accepted our invitation for today's precious guest lecture, which will be delivered by you. So before that, may I request you that you should proceed your guest lecture, and before that, please give some details about your journey, how you reached from India to Malaysia, so that our participants will know about how to build career there. Thank you, sir.
Thank you very much, Dr. Pranita. Can I share my screen now?
Yes, sir, please.
Is my screen visible?
Yes, sir.
Okay. Thank you very much for the nice introduction. Good morning, everyone. It's my pleasure to meet you all on this Sunday morning in the seminar organized by Sri Sivaji Educational College, Science College, and in collaboration with the Microbiology Society of India. Thank you very much to the organizers, the management of SSES College, the Microbiology Society, with a special mention of thanks to Prof. A.M. Deshmukh and the convener of this program, Dr. Pranita. Okay, so this is something which is I do every day. The title of this talk, which I say how to make microbiology teaching and learning interesting. So before going into that, actually she wanted a brief introduction of what I was doing all these years. So just a brief glance, like so this is my short journey, like how I ended up in Malaysia. No, actually, it's my passion was in teaching. So I started my career in teaching. In between, I went to industry. I was with different industries, starting with food industry, which is Taj Coromandel Hotel. It's a 5-star hotel in Chennai. I was a food microbiologist there. Then later, I went again into teaching, then shifted back again into industry. I was with the research and development centers of cosmetic industries, personal care industries, and pharmaceutical industries that were dealing with Ayurvedic drugs. So sort of a mixed experience, food, cosmetic, personal care products, and pharmaceutical products and whatnot. So, but my passion was always in teaching. So again, there was an opportunity which came from abroad to teach microbiology to medical and dentistry students. That's something new, like, because basically I'm a general microbiologist, though my PhD topic was on medical mycology. So it was quite interesting and challenging. So I took up this opportunity, and now almost 12 plus years in Malaysia. And I started again, see, because I was shifting between teaching and industry. So I had to start again from the scratch. When I came back to Malaysia, started as a lecturer, went into senior lecturer. Then I was a associate professor. Then there was a designation to which I was a senior associate professor, and now finally a professor of microbiology. So it's a long journey, but interesting. Like, what I want to tell you is, like, see, I did my BSc and MSc from a remote village, okay, in Tamil Nadu. So from that small or humble beginning, I started, like, then I was able to explore into the different opportunities. That's what as students you need to understand, that the opportunities are wide for microbiology. So I was with different industries, which I have already discussed, and then I came back to teaching. So microbiology is one dynamic field, like it's growing, and then now there is no boundaries. Actually, you don't study microbiology, you work in microbiology industry alone. So it's all microbiology, biotechnology, and all that. Everything has got a multidisciplinary approach. So there is no boundaries that you need to work only in a particular field. So with that brief introduction, like, let's jump into the topic of interest or the topic of talk today. which is also my passion, which I told you, like, so how to make it interesting is always a question. So teachers and the students, both of them, they face this challenge, like sitting in a class for long hours. Okay, and we always believe, like as the 21st century teachers, that teaching and learning should go hand in hand. It is not a separate entity. You teach and they learn separately. It's teaching and learning together. So, and then this is the era where it is a student-centered learning. So it is very, very important to make the teaching and learning enjoyable. It can be done only when the complex topics are made simple. It can be made fun-filled. Okay, so that's a whole idea of this talk. So this lecture is an attempt to convey my experiences, my thoughts as a microbiology lecturer. Okay, and thank you everyone for joining this event on a Sunday, okay, which is usually an off day or a family day. Okay, and I respect all your time here. Thank you for joining with me today. Okay, so these are the different methods what you can use to make this interesting. Usually, like when you start a class, you have a good opening for the class. Then you have the most of these, as teachers, you may be doing it. Use of cartoons, use of interesting correlations, use your creativity in teaching and learning. Okay, and use stories to teach them. Use of mnemonics to teach them. Okay, use interactive tools like quizzes, Kahoot. Okay, and then there are a lot of new teaching techniques which can be used in regular classrooms, like problem-based learning, flipped classroom, and so on. So I will discuss some of them for today's session. Okay, so now I have a topic, something like what I'm going to say is like, if I want to open a topic, so first I should make my students feel at ease. So I have a topic which I'm going to talk on the importance of the emerging fungal infections. So I use this slide. I just put the slide and ask them, what is this ship? So everybody will be I always believe in an interactive class. So they jump up and say, this is Titanic. And I ask them, what happened to this ship? Then they say, like, it sank. Then, okay. So before that, I also asked them, like, do you know, like, this movie Titanic got how many awards? They say, like, okay, I pose this question. Do any one of you want to attempt, like, how many attempts, I mean, how many Oscar awards this movie Titanic won? You can chat as well. Okay, so they give, when we talk of student, to talk to students, they give various numbers. They say 1, 2, 3, and so finally I tell them, like, what number of awards it got. So for all of your information, that's a movie which has got the highest number of Oscar awards. It won 11 Oscars. Okay, and it is shared with the other movie which is called Ben-Hur, which has also got the highest number of Oscars. So, but the topic is not about Titanic today. Then I say, like, this hit an iceberg. So what's the specialty of an iceberg? Then they keep on exploring their answers. Some of them come with really nice, funny answers. They say, like, it's made of ice. They say it's hard. And some of them say it's floating. Okay. But the point is, like, and one of them finally comes up with an idea, says, okay, no, this is an iceberg is something which is what you see in the top of the sea is just a small portion, and what is hidden below is just a major portion. Almost a 90% of thing is hidden. Then what I do is, like, just a minute, slide is not changing. Okay, so I just tell them that the importance of the iceberg, and then I relate this, that what the iceberg I'm talking about is not the iceberg which hit the Titanic, but I tell them it's a medical mycology iceberg which I'm talking. So which means, like, the number of fungal infections, what you actually see is very, very limited. It's just a tip of an iceberg. But what is not visible, it's a major chunk. That like the outbreak of mucormycosis and other things like which happened during the COVID-19 era. So this is how you when you start a class with something on a general topic, something related to a movie or something which they have already studied, they get to get the interest on the topic. So that's just to break the ice. You have this. Okay. Now, then you can always use some latest interesting headlines from the newspapers, okay, which is pertaining to the topic. So which actually helps them to keep the things more interested. Okay, so I always do this, like take some glimpses of pictures from the latest movies I put there and try to relate it with the microorganism which I'm talking about it on the day. Say this, if this is fungi, I say like I use this famous dialogue from Pushpa movie, which is a blockbuster hit in India. So I just say like, don't think fungi is something very simple, but it can cause life-threatening infection. Then I talk about what infections they cause. So, okay, now something like a basic microbiology topic, something where we talk about the differences between Staphylococcus and Streptococcus. This is something, and we teach them like how they appear in gram stain and what is the arrangement. That's a basic class we start with. But what I do for this class, instead of just telling that Staphylococcus is gram-positive cocci in clusters and Streptococci is gram-positive cocci in chains, I give them some interesting anecdote which they could remember and relate it so that they don't confuse this. So what I do is like, I give this example. I say, imagine there are two schools. Okay, there is a school which is very, very disciplined. Okay, very, very strict. Okay, and there is another school which is very, very casual and not formal. Participants, can you please mute yourself, please? Participants, I would appreciate if you can mute yourself, please. Okay, thanks. Right. So I just say that there are two schools which are there. Like one is a very enjoyable school, and another one is a very disciplined school. I compare the enjoyable school to Staphylococcus and the strict school to Streptococcus. What I say is like, oh, imagine that there is an assembly which is happening. So in an assembly in a strict school, what happens is all the children are required to stand in a line, okay, stand in a queue, okay, so stand in a proper alignment. So I say like, that's how Streptococcus is arranged. It is arranged in chains. Then I come to the other part and say, this is a fun school where students enjoy so there is no, they all clutter together, they all aggregate and stand as a group. So that's what happening in Staphylococcus, which is a cluster, okay, gram-positive cocci in groups. Okay, then I tell them, since it's a fun-loving school, okay, these students, other than studies, they participate in a lot of other extracurricular activities. So they win a lot of medals, which are, the gold medals, which are golden yellow. Okay, and then I tell them that Staphylococcus aureus produces a golden yellow colony, an yellow color colony in nutrient agar, and a golden yellow color colony in mannitol salt agar. Okay, so that's how I start. So I say there is no such thing here. And then when we come to the next one, I talk about the biochemical tests, which they need to know the difference between Staphylococcus and Streptococcus. I say that already I told this is a fun-loving school. So the students, they play with putting on bubbles. Okay, that's what I tell about the catalase test. I say like how catalase is produced. I tell the principle of catalase, how it is positive, how hydrogen peroxide is broken down to oxygen to get the bubbles. Okay, the effervescence in the biochemical. Okay, and then I go on to tell that this is a fun-loving school where they eat jelly, and then I talk about the coagulase test where after the coagulation reaction, then the agar gels. Okay, it becomes solid. So that's how. And then I say like these fun thing is not there in Streptococcus. So this is negative for the catalase and the coagulase test. that's how I relate it to that. So they also remember this. I also tell them that since it's a fun-loving school, okay, they are very simple, down to earth, so they can grow on a basal medium. And while this is very disciplined and principled, so they need a fastidious medium to grow. That's how I compare. So that's a major comparison between Staphylococcus and Streptococcus that they need to know. So at the end of this lecture, I'm sure like at least 80% will take back this memory, like, and then they never confuse which is the cocci in clusters, which is a cocci in chains. Okay, so other ways which you can do is like use of cartoons. So cartoon is always a fun, like, and then when you use cartoons which are related to science and medicine, then it's always interesting for them. So this is a dermatophyte skin infection of the foot, and then it's called tinea pedis. So, and then when I talk about dermatophyte infection, I say like, see, you can get these dermatophyte infection if somebody has got a tinea pedis, which is the infection of the foot. It can be transmitted by sharing a footwear with the other people. So I tell this, but to make it fun, what I'd say is like, I just put the slide and say like, you always say like, no, you want to be, don't judge other people. So when you want to judge other people, you'll always say like, you should be in their shoes, okay, to judge them. Okay, and I say like, that's a famous thing, like, and then say like, but before going into their shoes, make sure that they don't have an athlete's foot or the tinea pedis infection. So this is how I relate the cartoons. Okay, most of these cartoons not necessary we need to make. You just go into Google, just search that cartoons related to this topic. You may get nice cartoons. Sometimes you can create your own cartoons also. It's always fun also. So this is some cartoon which is already in the net, which I use, and how we relate it to our topic is that which is more important. Okay, now how do you correlate simple things which you have normally observed to your scientific remembrance? What I'm saying is like, say we all know that Vibrio cholerae grows on TCBS agar. It produces a yellow color colony. The color of the medium normally is green color, and then when Vibrio cholerae grows, it produces a yellow color colony. So if I say this, the students who are they have to memorize this that fact. Originally the medium was green in color, and then later it became yellow color. So they need to memorize this point. So what I do is I don't stop with this. I also say that you remember, like in your paddy field, the paddy or your crop is a green in color. It can be a paddy field, wheat field, whatever they have seen. I just tell them it's green in color initially, and then when it is ripe for harvest, it turns yellow. Same way, like when you try to grow Vibrio cholerae in TCBS, it changes from green to yellow. So now with this small comparison, this message which they take home, okay, that's very, very loud. So they remember this for a lifetime. Okay, so this is something from immunology, which is one of the most difficult lectures when we talk about the major histocompatibility structures. There is the molecule of MHC1 or MHC2, how they are different, and we compare the structural things. So this is one of the most difficult topics to teach in immunology. So, but you can make it more easier by putting little comparisons like what I told you. So what I do is like, say for example, this peptide that binds to the MHC1 molecule is a smaller peptide. The one which binds to the MHC2 is a larger peptide. Okay, so that part is okay. So, but this peptide, since it's a smaller peptide, then the anchor residues which hold them, it is only on the ends of the peptide, whereas it is to the entire length of this peptide. When I tell this, they all get confused. So what I use is a simple trick. Is I put this picture. I say like it's a small peptide, so it's enough that you can hold it at both ends of the peptide. So I compare it to a snake and say like the peptide. Imagine the peptide is a small snake, so you can hold it at both ends because it is small. But if it's a larger snake, I say imagine it's an anaconda, or if it's going to be a bigger python, then you need multiple people to hold it, right? So that's how it also works. Like in a when major histocompatibility molecule 2 holds a peptide, because it's a larger peptide, there is a need for more anchor residues. So when I say this and show these pictures, okay, they understand that much better than I just theoretically or tell them to imagine things, which actually it's very difficult for them to comprehend a difficult topic in immunology. Okay, so this is something on introduction to viruses. So we all know that these are the DNA viruses which are important as far as medically important conditions are concerned. These are the major viruses. So just to have this, I just related to the happy viruses. So I say like, but this happy is a little happy, and then I say like the HHAAA and sorry, APPVY is what? Herpes, hepadna, adeno, papawa, parvo, and pox. That's all. So if they remember that these are the viruses which are the DNA viruses which are medically important, that much is enough. So out of this DNA viruses, what you remember is like, out of this group, which is enveloped, which is non-enveloped, again they'll get confused. So I use this, I say herpes, pox, hepadna. I use this code as a high profile hat. So H for herpes, P for pox, and H for hepadna. So I say like a high profile hat. So when you put a high profile hat, what do? You try to hide your face, right? So it is enveloped. Okay, and then the other three which are there in the list is they are naked. So I say this is you remember the pap smear, and I tell them what is a pap smear and all that. I say these viruses which have the alphabets abbreviated as pap, they are the naked viruses among the DNA viruses. So, and then there's a very simple thing to remember in one slide, all the viruses and their structures also. I say all these viruses are isosahedral symmetry, but except for pox virus, which has got a complex symmetry, but all others are icosahedral. So this one again easy to remember is like, I say like, why pox virus? Okay, I say pox is within a box, so it's complex. So all others are icosahedral. So they carry this message and easy to understand rather than memorizing the whole list of which DNA viruses are enveloped, which DNA viruses are naked. Okay, so again, this is another organism which is called Cryptococcus neoformans, which is a fungi. And you know, like these are found on the pellets of birds, especially pigeons, and they can cause life-threatening diseases, including cryptococcal meningitis. So the fungi is actually, I use the latest references, what we get in the paper. I show them the leading journals with the latest references. Okay, just for them to glance, not for them to study, but just to get an idea like what is the importance of this organism. So, and these are some paper clippings of what happened about these organisms. Okay, now the comparison comes here. This is a type of staining which is called a negative staining, which used for Cryptococcus neoformans, where I tell them the difference between a normal staining and a negative staining. I say a negative staining is where the cells are not stained, but the background is stained. Then when this is a negative staining of Cryptococcus neoformans, it's a yeast form of fungi. So you have the yeast cell, it's a capsulated yeast cell. So you see the yeast cell and the halozone around the yeast, which is a capsule. I compare this to a moon. So I take them, I tell them like, have you seen a full moon day? And then they say yes. Okay, what do you see on a full moon day? A full moon, they say. Then on a, when you see the full moon, do you see a halozone around the full moon? Then they say yes. Okay, and then I show this picture also. I tell them, imagine that the moon is the yeast cell. Okay, and then the halozone, what you see is the capsule, which is produced by the fungi. So they remember the staining, negative staining, and the thing. So I always, when I conclude this, I say, okay, if you see a single full moon, if you see a single full moon on a dark sky, then you are seeing a moon on a full moon day. And I also say like, if you see multiple moons on a dark sky, then probably you are looking into the negative staining of Cryptococcus neoformans. that's how they remember it. And then when this question, which is an important must know outcome for them to know about this fungus, okay, they will remember this. Okay, so this is again about a fungus which is called Candida auris, which is the one of the emerging fungal infection, which has got a major implication because it's very, very life-threatening. Okay, it's one of the potent Candida member, okay, which can cause life-threatening infection. So I tell them, and then what is this importance and all. But to make it simplified, I compare this with the 3 properties of this organism. I say it has got resistance, resilience, and rapid emergence. So when I tell them it's just these 3 properties, they don't remember. So what I do is like I put a picture of RRR, which has got a different meaning actually, but because it's a famous movie, they will carry this message. So I just put this RRR. I say the first R is for the resistance of the fungi, second one is for the resilience of the fungi, and third one is for the rapid emergence. Then I explain all these 3 terms of what they happen with the subsequent slides. So I say I put a slide on the resistant pattern and then say like to which antifungals they are resistant and how dangerous it is. Okay, and then I also say like Candida auris is the only fungi which is listed as a threat list, but not because all others are only bacteria. Okay, but the major threats among the fungi, it's only Candida auris, which is the most prominent one. So I show that. So this is for the resistance. Then I say like, what about the next one is about the resilience. Then I give this newspaper article which was published a little back about Candida auris. This is an interesting thing. What they found is like a patient was admitted of Candida auris infection. He was in the hospital for 90 days. And at the end of 90 days, in spite of the treatment, the person died. So that is not a significant part. What is significant is like after the person died, what they did is like they tried to collect samples from the different places in the room where the patient stayed. They found that the walls, the bed, the doors, the curtains, the phone, the sink, the whiteboards, the poles, everything in the room, including the tiles, okay, which were all contaminated by the virus. So, and then they went on to a level that you have to rip off certain tiles from the room to replace them with new tiles to remove this fungus. Such a recalcitrant fungus it is. Then I say this is the resilience property of Candida auris. Okay, and then I talk about where it is distributed, and then now say after February 15, 2021, this map which is updated by CDC is no more updated on where this Candida auris is present, because after the advent of COVID-19, it has come to all the countries in the world. So this that is for the rapid emergence. So these are the points. So I relate it to the movie RRR, and then I tell these stories so that they catch up attention on what we are trying to see. Okay, so again, this is again another fungus, which is a dimorphic fungus, which can exist in 2 forms. So I compare it with the cartoonic character or the Marvel character Hulk. Okay, Hulk can exist as 2 different characters as Professor Banner. Okay, and then when he becomes angry, then he becomes Hulk. So I say like the dimorphic fungi can exist both as a yeast form and a mold form. So when it is when Hulk becomes angry, then he changes the form. So I say like similarly, like when this dimorphic fungi is the temperature is changed for the incubation of the dimorphic fungi. Okay, it converts from the mold form to the yeast form. So I compare this with the Hulk, so that they remember that there are 2 forms, okay, of this fungi. Okay, and then like when we start teaching the streaking in the plates, when we talk about the isolation techniques of the quadrant streak and all that, we just make it more monotonous by asking them to do a normal streak or isolating a pure culture and all that. But why can't we make it more fun-filled? Okay, when we do a streaking experiment on the day, then you can also give them 1 extra plate and ask them to streak something which is creative. Okay, a picture or something which they can do. Okay, and then when you'll be amazed to see like how students can be so creative and all that. This is some examples of the agar plates which are steeped with colorful bacterial cultures. So that is something which we teach as teachers. We need to explore that instead of just monotony in teaching. Just we finish one experiment on sticking, then we take it to a different level to make it creative, so that they feel that microbiology or learning microbiology is a real fun. And then whenever it comes to the hemolysis of on a blood agar, okay, we just tell them alpha, beta, and gamma, and then stop with that. So, but what I usually do is like when I talk about teaching hemolysis on a blood agar, I tell them to write their name, okay, with the on the blood agar. So the next day, when the hemolysis has happened, they can read their name on the blood agar. So that will be really fun. Okay, so they will be more interested to do a laboratory technique or come to a microbiology lab with to thinking that it's going to be fun that day to learn a new technique, as well as to practice something like writing their name on a blood agar. The other way which we can do is like to create stories. Okay, to when we teach about some organism. This is one organism which is called Sporotrix schenki, which is a fungi which causes a disease called sporotrichosis. It is a subcutaneous infection on the skin. Okay, so and it's such a dry topic, and then usually how this infection comes is like these fungi can be on the rose plants. So whenever the gardeners or florists, they pick up the flowers, they get pricked by the thorn, and then they can gain entry of this fungus, and they can cause a subcutaneous infection. That much is only the information. So you tell them this is more dry for them to remember. So what I make, how I make this interesting is I tell them like a story. I say like remember like Schenki is the name of a girl. Okay, the Sporotrix schenki. Okay, just take this Schenki and say, imagine Schenki is a girl. Okay, and then say like I compare Schenki with Shuri, which is a Marvel character. So I just say like Schenki is a beautiful girl. So there is a boy who wants to propose to this girl. So how do you propose is with a rose. So I say when you think of love, you think of a rose. Okay, so and when you think of a rose, what else you think? You also think of a thorn. So I tell them like love always is not very nice. It also hurts. So think of a thorn. So when you're trying to pluck a flower, okay, which happens to the florist, they get accidentally pricked by a thorn. Okay, so that is how the mode of entry of this fungus into your body. Then I tell them that see, this is a culture of Sporotrix schenki. This is how it grows. Okay, a gray and a white colored colony. Okay, I keep this reference of Shuri and say like she has the tribal makeup all over her face, which is white dots on a darkly pigmented skin. So which is almost similar to the Schenki's colony morphology. Then I say like somebody who is in love, okay, you will see the stars in their eyes. Then what are these stars? Actually, I compare them that these are the asteroid bodies which are produced by the fungi, and then you can see them in the tissues. So I relate the asteroid body. Aster means what? Star. Okay, so I relate the asteroid body to the this is a diagnostic feature of Sporotrix schenki. So I relate it to the stars in the eyes of the people who are in love. So this concept, okay, after I tell that, they remember this, and then to make it more interesting, I also put a word about the treatment. I say Schenki, the last letters is Ki. Actually, incidentally, potassium iodide is a treatment method for the skin infected with Sporotrix schenki. So the formula for potassium iodide is Ki. So I just say that, and then this is how I create it as a story, so that the students remember this story for a long time. So whenever they finish their course, they come back after a few years also, they come and say like, we still remember the Shenki story, sir. So that's how the learning has to be fun. Okay, so again, these are the floral arrangement. This is how the spores are arranged. We say daisy arrangement, and then I show them the flowers, sporothrax. Okay. Now, the other point which I can stress is like the use of mnemonics. The use of mnemonics in teaching. So whenever you finish a topic, it can be on any organism. It can be on Streptococcus, Staphylococcus. It can be on Vibrio or whatever. So when I finally conclude, I use this mnemonics to create a memory bank. So what I do is like I just picked up Venom, which is a famous movie character. Okay, and then I put Venom. The idea of choosing Venom is like Vibrio also starts with B and Venom also starts with B. So I put that. And then, like Vibrio also produces the toxin, so Venom. Okay, and I show this all these points which are very, very important for them to remember. So Vibrio is oxidase positive, which they need to know, which is gram negative, which they need to know. The motility is darting motility, which they need to know. Okay, and then I talk about if they know more, because these students see a lot of movies, they also remember the characters of the movies. So I say like the sticky tongue of the Venom, I compare it to the biochemical test, okay, where you can use sodium deoxyfolate to mix it with the culture and produce a sting. Okay, and this the sting test. I compare the sting test to the sticky tongue of Venom. Okay, so same way we can do different organisms to different characters. This is all I picked up from Marvel. Okay, I used Hulk for Helicobacter. Okay, and then the reason, there should be some little comparison also. Helicobacter actually destroys the intestinal layers, and then it causes ulcers. So Hulk, when he is angry, also he keeps destroying things. So I compare that destruction of the tissue layers to the destruction of the buildings. Okay, and then I try to relate them to these mnemonics. Okay, so this is again another example of the character Captain Marvel to Campylobacter. That Campylobacter has got a seagull-like appearance. We always talk about the seagull appearance of Campylobacter. So I put this picture of Captain Marvel. I say it's sort of a flying position like a seagull. Okay, I just keep this as a reference for them to remember that this particular organism has got a seagull appearance. And again, this is Dr. Strange or Stephen Strange. I compare it to Salmonella or Shigella. So that's how I keep revising the topics which is already completed using a mnemonics. And then for mnemonics, you use some important characters from the movie, okay, or from the cartoons which they like, okay, so that they remember it for a long time. Okay, just to conclude, I just want to finish with an interesting news. I do not know how many of you know that there is a museum in Amsterdam, Netherlands, which is called Micropia, which is a microbiology museum. Okay, I haven't visited it, but one of my colleagues visited it. It's one of the fantastic places which every microbiologist should visit. So this one, like, it has got a lot of interactive stations. As you go in, like, your body will be scanned and you will be told like how many microorganisms are there on your skin. Okay, there is a place where you can just give a kiss and they'll say like in the kiss you transfer so much of microorganisms. Okay, so those are things which are fun loving. But what I'm saying is like other than your regular teaching, don't just stick on to your outcomes. You talk something about some general things about microbiology, what is happening in the world. That's more important for the students to know. Okay, so these are some pictures from the Microbiology Museum, which was taken by my colleague. Okay, and it's a dream destination for all microbiologists. But I wish like it is possible to create such a museum in India also. Like we have our own the strong player, High Media, which is a major group. I know, like our Microbiology Society is also having collaboration with High Media. They can work on something to create a Microbiology Museum, okay, in India, which can be a reference, which means like all organisms, their growth structures, okay, which can be created, and then it can be a wonderful place for us to visit. Okay, so that's all from me for today. I come from this university, Manipal University, Malaysia. Okay, which also which actually basically it's a university which has got MBBS and dentistry programs, and I'm teaching for dentistry and MBBS. Okay, and then if you have your friends or any of your colleagues who have their sons who want to do medicine abroad, okay, they can consider, which is one of the best universities in Malaysia, Manipal University College, Malaysia. Thank you, and that's it.
Yes, sir. Thank you so much, sir, for a very interesting and really very much informative session, sir. Actually, you have thoroughly given the idea that how to make teaching and learning very much interesting and effective. So from students' point of view, you have given lots of examples of the marvels and superheroes, and that is really a very good technique, sir. Lots of messages are there in the chat box too, that this idea is very much appreciating for giving some idea to the students is for teaching purpose. So really, sir, this way of teaching and learning is really very much appreciating from all the participants, and we also will try to learn this technique, sir, because what happens now, some topics are very much difficult to students. Most of the times they are unable to understand, but this is very good method that they will understand and they will like the subject. Means to love our subject, to for the development of our subject, it is very much important, sir. So thank you so much for a very nice guest lecture as well as the presentation, and yes, the information which you have shared. So I'm requesting all the participants, if they have any questions or queries, they can please ask or put it in the chat box.
First, I request speaker to type his mail in the chat box. So at the end of this session, participant can communicate personally on the mail.
Yeah, I'm doing it right now.
Thank you. Thank you.
Yes, sir.
If
Thank you so much, sir.
Yes, if all are appreciating your lecture.
Thank you, sir.
Yes, yes, and asking to repeat me that you, sir, you should repeat this lecture again and again. So
Sure, sir. Sure, sir.
yes.
Okay, maybe in a different geographical location, like if someone is organizing, I can repeat this lecture again.
Correct, correct.
Different audience, yeah.
Yes. So anyone want to discuss anything, please come forward.
There is one message in the chat box, sir, for you. What was the source of inspiration to use this novel method of teaching?
No, actually, it's always a challenge, no? Like when we go and teach in a class, like our MBBS programs, or we have a students of over 100 in a class. So to satisfy the 100 students and keep them in attention, like all the time, like for a 1 hour class, it's always a challenge. So I was always keep thinking like how to keep the class interesting. And I had some of my microbiology teachers who were great inspiration to me, and then they are also like one of the reasons, like for me to think out of the box on how to teach. Say for example, my one of my microbiology professors, like he used to compare the electron transport chain. He will say like imagine that why you need to have a lot of carriers, and then why it is carried from one carrier to the other carrier. Why not from first carrier to the last carrier you produce energy? Then he says, in a very simple example, he says, imagine there is a machinery work which is going on, and then each one is passing a brick. Okay, you are passing a brick across a place. There'll be 10 people standing and throwing the brick to each other, and then finally it reaches a place where the brick has to be used. Okay, so he says, instead of this, imagine like a person from the second floor throw a brick to the ground floor. Will the other person standing down, will he be able to catch? No, he will break his hand. Same is the level of energy. If there is no different carriers happening, okay, from the first carrier to the last carrier, you just throw the brick, the cell will not be able to, will not be able to harness the energy which is generated. So it has to pass through a series of carriers, okay, electron carriers. So finally the energy is produced. So it's just a simple example which is given by my professor when he taught me. So this is one thing which I always think like. Then I always think like, why can't we think something out of the box? So I used to think like, most of the times I keep thinking like, whenever I see a movie or whenever I see something new, I always think how I can relate this to microbiology.
Yes, definitely. That is appeared in your presentation. One more question is there. Sir, do you feel virtual 3D video sessions can be new teaching method in near future?
Yes, true. It is already used, like virtual 3D versions. Videos are used currently. Like even we use in our medical. It is good, like especially in a medical university where you have to study the internal structures, the functioning of an anatomy of an thing. Like you use of this interactive. We have instrument which is called anatomage. So you can just open each layer of the body and see what is inside, like with which is a virtual software which can be done. So same way, like when we talk about the different pathogenesis of microorganisms, like how they go into, say, for example, an organism which can capture the macrophage, travel in the macrophage to the different parts of the body and all that. So if it can be visualized by a 3D animation, that will be wonderful. That will be very easy for them to understand rather than we telling them orally. Yeah, that's a future for teaching. Yes.
Yes, sir. So one question is from my side. Maybe this is the question from the participants' mind also. That how we can make the experimental work, means practicals, when we are teaching to the students in laboratory. Then what is the effective method to teach them the practicals?
No, thing is like, see, practicals is like hands-on skills. So what they need to do is like not just completion of the practical, like when they learn a skill, be it teaching, be it just a streaking or an antibiotic sensitivity test, each one we should make sure that each student has learned the technique thoroughly. And then like when we incorporate the laboratory tests, in the curriculum, we should also see like what is the requirement in the industry. What sort of tests are usually done in a hospital, or what sort of tests which is done in a food industry or a pharmaceutical industry. So those things, if they are addressed as a practical, then it will be very easy for the students when they go out. Say, instead of just saying like you do this analysis, like you just say like a bottle of mineral water is given, how will you go about the microbiological analysis? That should be your experiment. So when they finish their thing, if the student is going into a water industry where he is going to be a quality control microbiologist, he should know like how to take the water sample and how to proceed with the test. So that will be more application oriented for the industry. Yeah.
Okay. There is one question from the audience, is that how can we implement interactive method or such methods where we use only chalk and board?
Chalk and board, actually, like teaching can be any initially it was chalk and board. Then when we started moving into the PowerPoints, the lectures where you have necessary provision, people use PowerPoint teaching only. No, but it is always now when we go into again small group teaching, like problem-based learning and all, they have also again gone back to the chalkboard method. Instead of the chalk, it has become a whiteboard where erasing is more easier. So it has become a whiteboard method. So when you write something on the board, rub and then rewrite and all that, no? When it comes to interactive learning, what I do is, like I was telling you the difference about Staphylococcus and Streptococcus. I don't teach the entire thing. I put this picture and ask them to relate. Okay, I ask two students to come into the front and then ask them to how they are going to relate this picture to the biochemical test. I just give them the clue that this picture is to relate to the biochemical test. If they have a background of the biochemical test of what is to be done for Staphylococcus, they know it's a catalase test. So they will think through and then they will come up with the answers. That's a way to make it interactive. Yeah.
Okay. There is one more question, that what strategy do you implement to reduce the performance fear while performing the practical?
No, the thing is, like, see, students will make mistakes. Okay, so we should appreciate that. We should not be harsh to the students. I would say, like, if when we teach streaking, if my student does it on the first streak itself, he does a quadrant streak very nicely, I appreciate and tell them, like, when I was a student, I took 10 times to learn a proper sticking. I'll tell them, like, I used to plow on the agar plate instead of doing a nice sticking. So that's a way we learned. So I tell them, like, to make mistake, the students should not be intimidated. Like, when they make a mistake, they should take this as an opportunity to learn. Okay, so just encourage them, because not every student, say if you have a gram staining practical, not everybody will be able to do the practical successfully. Some of them may not get the staining properly. Some of them will not be able to focus on an oil immersion lens that day. Okay, give them practice. You can repeat the experiment one more time. Okay, and then they will be able to achieve it. Yeah. Different students have different pace of learning. So some are very fast, but we cannot keep that as a gold standard. Some are very slow learners. They need time to work on.
Yes, there is one message for you, sir. Nice discussion on remembering concepts. Participative learning is being encouraged in national education policy. If teachers can initiate the discussion, the recollection of the topics taught can be done in an interesting manner. For example, draw a spider, write the initiating sentence in the center, and ask the students to write the keywords on the extensions.
That's correct. Actually, like, this is almost similar to the PBL, which is the problem-based learning. We just give a one-liner or a scenario, and then the students will be asked to brainstorm and come up with their own ideas. Okay, which they can put in a whiteboard. Okay, and then they can group discuss. You give them time also. You just, sometimes what we do is, like, we just give them a scenario. Okay, and then we give them 1 hour time, which can be a library time. They can go to the book, read, come back. And then after that, there is a brainstorming session. They can come up with the ideas, and then all the ideas are put together in a whiteboard. And finally, we say, like, what are relevant and what are not relevant. That's how we conclude the lecture. Yeah.
One question is, so what is the importance of drawing skill in microbiology?
Basically, what you need is a schematic drawing, not a neat picture. Yes. But at least you should know a schematic drawing. Okay, so a cocci should look like a cocci, not like a bacilli. So that, if that level of drawing is there, that should be fine.
Oh yes, sir, definitely. Sir, as per the postgraduate students, one question is from my side, is that we are having a postgraduate courses in microbiology here, and they are having projects, small projects or dissertations as a part of their curriculum. So what is the thrust area, on which areas we should give the topics to the students, so that they will get benefited after their post-graduation to acquire jobs or their research?
The one thing is, like, whatever topics, it also depends on not just topics, the expertise of the supervisors. If the supervisors are have an expertise on bacteriology or virology or mycology, depending on their thing, they can choose the topic, so that the supervision will be much better than choosing topics which they do not know. But I told you, like, it's a multidisciplinary approach now. It's not solo. So if the guide is not an expert also, he can have co-supervisors, 2 or 3 from other places as well, from other university, Okay, other it can be from local, abroad, because now everything is online. Anything can be discussed online also. So you get ideas from them and then you can implement. But what is necessary is like the student should know like what area he wants to work, depending on what he plans to go in future, whether he wants to go into medical microbiology, or he wants to be in food microbiology, or he wants to be related to pharmaceutical microbiology. So based on this, he can choose his topics. So that will be a sort of a project which will be helpful for his job also. So that's one thing. And the trust areas are already there. Like, see, if you see, like every country will have a science and technology council, which they list the trust areas, what is important for the industry's point of view, what is important for the humanity point of view or the community point of view. And then we need to choose topics which are very relevant. So I always feel like, but sometimes no, if everybody goes and research only for looking into the latest and the recent developments, and who will do the basic research? That's also important. Like if you look into my PhD topic was on fungi, medically important fungi, but I was looking and studying certain evolutionary aspects of the fungi, which is totally different, which is not a current area and all that. But that's also adding to the knowledge. So it depends on, but idea is like you have to come up with something new to the society, which will be useful to the society.
Yes, sir. Thank you so much, sir, for satisfying the queries from the audience. Really, it is very good technique of teaching, and so that learning from the student side get very much effective for that. If anyone has any query or question, they may put in the chat box.
There is one question which says, which topic in microbiology is most difficult to teach?
Yes.
That is because whatever topic you are comfortable with, yeah, you feel it easy. Whatever topic you are not comfortable with, you feel it is difficult, because say, for example, if you ask a basic microbiologist to teach a topic in medical microbiology, he will find it difficult because that is a little away from his regular topic. But only when you start exploring and start reading on that particular area, you will find that that is also simple. Okay, I'm a very good example because I did general microbiology. Okay, but I am teaching medical microbiology, so which is very deep into medical microbiology, because I'm teaching the medical students so about the diseases and the complex pathogenesis and all that. So which is actually not of my part of my curriculum, like when I studied also. So the idea is like you need to learn. Teaching is like lifelong learning. Okay, any thing is lifelong learning, but teaching definitely is lifelong learning. We need to be updated. We need to keep reading on the latest things, what's happening. So only then we will be able to cope up to the level of students, because students are reading every day. Okay, if you have some good students in the class, they will be more informed than us. Then how to beat them? Beat them in the sense, beat them in the knowledge. Like, so you want your student to listen to you, you should know more than what your student knows. Okay, so that way, like you have to learn. That's no other goal.
Yes, sir. Definitely. Oh, thank you so much, sir. Yes, sir. Dr. Deshmukh, sir, shall we conclude here now?
Yes.
Yes, sir.
Let me again reiterate that today's lecture was slightly different from the normal lectures.
Yes, sir, definitely.
And people, all teachers have appreciated. We have recorded it. We'll circulate on YouTube.
Thank you, sir.
And I'm sure more and more teacher will take benefit in the future also.
Sure, sir. Sure.
Thank you. Thank you, sir. Thank you, Pranita.
Thank you very much, sir. Thank you for the opportunity.
Thank you so much, sir. Thank you, Sabesan, sir, on behalf of Organizing Committee of Shivaji Science College, Nagpur, Honorable Principal Professor Dhore, sir, and Honorable President of Microbiology Society India, respected Dr. Deshmukh, sir. On behalf of both this organization, I am very much thankful to you for such a precious guest lecture from your side, sir. Thank you so much.